Today we will discuss about glycogen breakdown, that is glycogenolysis, and glycogen synthesis, that is glycogenesis. Now, glycogen is a readily mobilized storage form of glucose. It is a very large branched polymer of glucose residues. Most of the glucose residues in glycogen are linked by alpha 1,4 glycosidic bonds. Branches at about every 10th residues are created by alpha 1,6 glycosidic linkage. The presence of glycogen greatly increases the amount of glucose that is immediately available. Glucose is virtually the only fuel used by the brain except during prolonged starvation. Glucose is present in the cytosol in the form of granules. These granules contain regulatory proteins as well as the enzymes that catalyze the synthesis and degradation of glycogen. The pathway of glycogen breakdown was elucidated by the studies of Karl Corey and Gerty Corey. Glycogen phosphorylase catalyzes the sequential removal of glycosyl residues in the form of glucose 1-phosphate from the non-reducing end of the glycogen molecule. Glucose 1-phosphate released from glycogen can readily be converted into glucose 6-phosphate. This phosphorylation is catalyzed by the enzyme phosphogluconatase. Now, the phosphorylated cleavage of glycogen is energetically advantaged because the released sugar is phosphorylated. In contrast, a hydrolytic cleavage would yield glucose, which would have to be phosphorylated at the expense of an ATP to enter the glycolytic pathway. An additional advantage of phosphorylated cleavage for muscle cell is that glucose 1-phosphate is ionized under physiologic conditions, therefore cannot diffuse out of the cell, whereas glucose can. Now the glycogen is degraded to a limited extent by glycogen phosphorylase alone. However, the alpha 1,6 glycoside bond at the branch point are not susceptible to cleavage by glycogen phosphorylase. It can only remove or only catalyze the glycogen alpha 1,4 linkage. So glycogen phosphorylase stops cleaving alpha 1,4 linkage when it reaches a terminal residues 4 away from the branch point. The action of glycogen phosphorylase on two outer branch of a glycogen particle is shown in this figure. 5 alpha 1,4 glycosidic bonds on one branch and 3 on the other branch are cleaved by phosphorylase according to this schematic diagram. Cleavage by phosphorylase stops at this stage because terminal residues, which are colored in the blue and yellow, are 4 residues away from the branch point. So a new enzymatic activity is required. A transferase, that is glucan transferase, shifts a block of 3 glycosyl residues from one outer branch to the other. The alpha 1,4 glycosidic link between blue and green residues is broken and a new alpha 1,4 glycosidic link between blue and yellow residues is formed. This transfer exposes green colored residue to the action of another degradative enzymes, that is alpha 1,6 glucosidase, which is also known as the debranching enzyme. This enzyme hydrolyzes the alpha 1,6 glucosidic bond hydrolytically, resulting in the release of glucose residues. Now we will discuss about the glycogenesis, that is the synthesis of glycogen. 3 lines of evidence indicated that glycogen is synthesized in vivo by a pathway different from that of its degradation. UDP glucose is the glucose donor in the biosynthesis of glycogen. UDP glucose is the activated form of glucose. Now, UDP glucose is synthesized from glucose 1-phosphate and uridine triphosphate in a reaction catalyzed by UDP glucose pyrophosphorylase. This is the schematic diagram of reaction where UDP glucose pyrophosphorylase catalyzes the formation of UDP glucose. New glucosyl units are added to the non-reducing terminal residues of glycogen. The activated glucosyl units of UDP glucose is transferred to the hydroxyl group at the C4 terminus of the glycogen to form alpha 1,4 glycosidic linkage. This reaction is catalyzed by glycogen synthase. Glycogen synthase catalyzes only the synthesis of alpha 1,4 linkage. Another enzyme forms alpha 1,6 linkage that make glycogen a branched polymer. A branch is created by the breaking of alpha 1,4 link and the formation of alpha 1,6 link. This reaction is catalyzed by branching enzyme. The block of residues, typically 7 in number, is transferred from the non-reducing terminus and it comes from a chain at least 11 residues long. In addition, the due branch point must be at least 4 residues away from a pre-existing one. Glycogen synthase can add glucosyl units or glucosyl residues only if the polysaccharide chain already contains more than 4 residues. That means glycogen synthesis requires a primer. This priming function is carried out by glycogenin. It is a 37 kilodalton protein bearing an oligosaccharide of alpha 1,4 glucose units. The first unit of this chain is covalently attached to a specific tyrosine residues of the glycogenin protein. Actually, glycogenin autocatalyzes the addition of some 8 glucose units and UDP glucose is the donor in this autocatalyzation or autoglycosylation. After this, glycogen synthase, which is tightly bound to the glycogenin, takes over. So the elongation stops when the synthase is no longer in the contact with glycogenin, which forms the core of the particle. So this is all about the glycogen breakdown and glycogen synthesis. Thank you.